Welcome to 3M Webstream. I'm here with Dr. John Hester. He's a research scientist in 3M Purification. John is going to talk to us about a new product 3M is bringing out in the field of clarification. The product is called Emphaze AEX Hybrid Purifier. John is going to talk to us about the product design, its performance, and the value the product is going to deliver to our biopharmaceutical customers in their purification space. Welcome to 3M Studio, John.
Thank you very much, Himanshu. I'm glad to be here, and I'm very excited to talk about our new product. At 3M, we pride ourselves on providing best-in-class clarification products that are widely used to clarify cell culture fluids prior to chromatographic purification of life-improving protein-based therapeutics. Our Zeta Plus depth filters, available in grades to meet a variety of needs, from direct mammalian cell culture harvest applications to centrates or lysates, have led the industry in bioprocessing clarification for years. The Zeta Plus encapsulated system provides the gold standard in bioprocess efficiency with fully single-use capsules and a space-saving ergonomic design. The Emphaze AEX Hybrid Purifier represents the next technological step forward in biopharmaceutical clarification. This product is an all-synthetic clarifying device that, for the first time, brings substantial ion exchange functionality to clarification. It is also the first ever clarifying device with a defined mechanical pore size. The extremely high anion exchange capacity of this all-synthetic media construction provides reduction not only of the cell debris that contributes to turbidity, but also soluble anionic impurities like DNA and a significant portion of host cell proteins. Those impurities are generally removed by chromatography columns further downstream in the purification process. But by building that same kind of chromatographic functionality into this next generation clarifier, we can strongly reduce them way up here. Additionally, the defined 0.2 micron pore size of the Emphaze AEX Hybrid Purifier enables it to provide unsurpassed fine particle and turbidity reduction throughout the entirety of product clarification, and even greater than 6 log bioburden reduction. I believe this new product is going to define a step change improvement in biopharmaceutical clarification, especially for monoclonal antibody production where protein A capture chromatography is used. By providing better product purity upstream of the protein A chromatography column, we've shown that you can not only better protect that critical unit process, but dramatically improve its performance as well.
This sounds very interesting, John. Can you tell us more about the design and development of this new product?
Well, the development of this product began the same way I think all of our most successful new products do. It began with a close relationship with our customers that taught us something about their unmet needs. In this case, our customers told us they wanted all synthetic clarifying devices with well-defined ingredients and highly predictable performance and extractables profiles. From there, it required the application of unique technology from 3M's corporate labs. In fact, this product would not have been possible without consistent technology investments and persistent investigations by our friends in 3M corporate research in 4 separate areas: nonwoven media, surface modification, advanced materials, and highly asymmetric membranes. Finally, the development of this new product involved an aspect of our most successful products that we can usually only hope for: new technology creating a solution to perhaps unanticipated needs. In short, our efforts to create an all-synthetic clarifying device, we doubled down on anion exchange functionality as the primary mechanism for purification. This graph, a comparison of the anion exchange capacity of the Emphaze AEX Hybrid Purifier with that of a conventional depth filter, illustrates the high level of anion exchange capacity I'm referring to. Conventional depth filters rely on a variety of adsorptive mechanisms: interactions between impurities and cellulose, filter aids like diatomaceous earth, as well as amine polymer additives to remove product impurities. The Emphaze AEX Hybrid Purifier really relies on only one adsorptive mechanism, an extremely high level of anion exchange functionality. This makes it a simpler device in some ways. But when we and some of our collaborators started experimenting with clarifying media having this level of anion exchange functionality, we were really excited by the results we could achieve, not only with cell debris, but with the soluble impurities conventionally removed only by downstream chromatography steps. Here, I'll show you the construction of the Emphaze AEX hybrid purifier. We call it a hybrid purifier because it contains two media types, providing two modes of purification, anion exchange and mechanical sieving. First, the Emphaze AEX hybrid purifier contains 4 layers of a quaternary ammonium, or Q-functional, nonwoven, a proprietary surface modification technology developed in 3M's corporate labs has enabled the functionalization of a U.S. pharmaceutical-grade polypropylene nonwoven with this covalently attached cationic polymer. In fact, this Q-functional polymer accounts for roughly 2/3 of the mass of the functional nonwoven, and it's this very high level of functionalization, we call it hyperfunctionalization, that provides the high anion exchange capacity of the product. Second, the Emphaze AEX hybrid purifier contains a very asymmetric, high-capacity polyamide membrane. This membrane is the deepest and most asymmetric membrane we've ever commercialized as part of any product. It contains 9 separate zones of pore size, spanning a roughly 20x range from top to bottom and terminating in a 0.2 micron pore size. This provides a defined mechanical pore size for the sieving of fine particles, as well as greater than 6 logs of bioburden reduction. It also provides excellent protection of a downstream sterilizing membrane. The Emphaze AEX hybrid purifier will be available in our popular family of fully encapsulated device formats, spanning device sizes from lab to production scale.
The new product appears highly differentiated compared to conventional clarifying products. Can you comment on its performance also?
In fact, its unique design gives it very different performance than conventional clarifying products. I'll show you some example data. This is data from the clarification of a Chinese hamster ovary cell centrate having an initial turbidity of about 84 NTU. We clarified the centrate using both the Emphaze AEX hybrid purifier and one of our highly charged fine grades of a conventional depth filter to compare the performance of these two types of clarifying devices. In this figure, you can see the pressure for each device in the bottom graph, the conventional depth filter in blue and Emphaze AEX hybrid purifier in green. Outlet turbidity is shown in the top of the graph. Both of these excellent clarifying devices had very good throughputs with this fluid, greater than 600 liters per square meter. In general, we see that the Emphaze AEX hybrid purifier exhibits throughputs similar to a fine grade of a best-in-class conventional depth filter, like this one. The turbidity reduction of the Emphaze AEX hybrid purifier is better than the conventional depth filter and consistent throughout the entirety of the run. This is the power of the defined outlet pore size of 0.2 microns. Where you really see the differentiation of the Emphaze AEX hybrid purifier is in the reduction of soluble contaminants. This figure shows reduction of DNA during the same clarification run. As you can see, this highly charged conventional depth filter provided excellent DNA reduction at the beginning of the run, but the DNA reduction dropped throughout the run as its relatively moderate anionic charge capacity was exhausted. By contrast, the Emphaze AEX hybrid purifier removed greater than 4 logs of DNA throughout the entirety of the run. This is a typical pool DNA reduction we observe for a wide variety of fluids. I'd also like to highlight the hockey stick shape of the Emphaze AEX hybrid purifier's pressure curve during this run. This is characteristic of the Emphaze AEX hybrid purifier and is due to its unique design. As the anion exchange capacity of the Q-functional nonwoven nears exhaustion at the end of the run, fluid impurities pass through to the membrane. This results in a characteristic membrane plugging curve at the end of the run. In most cases, this provides the product with a fail-safe feature by guaranteeing a pressure endpoint prior to breakthrough of soluble contaminants like DNA. So in most cases, you don't need to wonder if soluble contaminants like DNA are breaking through at the end of the run. Finally, this graph shows host cell protein reduction by both clarifying devices. Again, the Emphaze AEX hybrid purifier substantially outperformed the conventional depth filter due to its extremely high anion exchange capacity. We generally observe between about 20 and about 40% HCP reduction by Emphaze AEX hybrid purifier, depending on the isoelectric point distribution of the proteins that are present.
So John, is it fair to say that this is a novel solution for clarification?
Yes, it's definitely a breakthrough technology in clarification. There's really no other clarification solution with either this high level of anion exchange functionality or a defined mechanical pore size. Here we're doing flow-through chromatography and bioburden reduction simultaneously with clarification. One thing that's really quite striking is to look at a comparison of the particle size distributions of CHO centrates clarified by conventional depth filters and the Emphaze AEX hybrid purifier. Here we use dynamic light scattering to look at the particle size distributions of a CHO centrate and then samples of that centrate that have been clarified by two different grades of conventional depth filter, a coarser one in red and a finer one in blue, and the Emphaze AEX hybrid purifier in green. Keep in mind that this type of light scattering tells you about the distribution of particle sizes in a sample, but not the number of particles. Also, it's most sensitive to large particles that scatter light more strongly, so you often won't see small particles when larger ones are present. In the centrate in black, we see relatively large particles consistent with cell fragments. Smaller particles like soluble proteins are of course there, but we don't see them because the light scattering is dominated by the large cell fragments. In the fluids clarified by the two conventional depth filters, those cell fragments have been removed. We see a bimodal distribution of particle sizes, one at about 100 nanometers, consistent with the size of DNA and protein aggregates, and a second one at roughly 10 nanometers, consistent with soluble proteins. In the case of the finer of the two depth filters, there's a relatively smaller proportion of the aggregate-sized particles. But what's really exciting is the particle size distribution of the fluid clarified by Emphaze AEX hybrid purifier. Even though this light scattering technique is most sensitive to larger particles, we see only particles in a size range consistent with soluble proteins. In fact, we can't see any particles larger than 30 nanometers, or 0.03 microns. I think this is a striking illustration of the step change in clarification performance provided by the Emphaze AEX hybrid purifier's unique all-synthetic construction. In addition, this shows why the product provides such excellent protection of downstream sterilizing grade membranes. In fact, this product provides fantastic protection even of 0.1 micron sterilizing membranes, like our new LifeAssure PDA010 sterilizing membrane. This should enable routine mycoplasma level protection of the downstream process.
I can definitely see, John, this is a differentiated product in terms of its performance during clarification, particularly with respect to DNA and host cell protein reduction. It looks like essentially we are doing a flow-through chromatography during clarification.
Yeah.
Can you elaborate the benefit of you doing chromatography during clarification? Because the protein A or affinity chromatography done during monoclonal antibody purification is supposed to remove most of the DNA and HCP. So would you be able to comment on the performance of Emphaze AEX hybrid purifier benefiting this process?
Right. Well, look, we've all seen TV commercials where someone loads a complete cake or a pizza into a dishwasher. They run the dishwasher and show that it's capable of making the plate with the cake on it pretty clean. This image makes it an effective advertisement and highlights the excellent capabilities of modern dishwashers. But I don't believe most of us routinely load the dishwasher with cakes and pizzas. After all, the dishwasher is a critical unit process in most of our kitchens, and we would expect that over time a practice of loading unnecessary food into the dishwasher would eventually affect its cleaning performance and result in increased maintenance requirements. My own family has a pretty nice dishwasher in our kitchen. In fact, I've seen a YouTube video of the cake demonstration done on the model we own. But I can say from experience that if I inspect the dishes carefully when we unload them, I can clearly see a relationship between the food load on the dishes going in and the cleaning performance of the dishwasher. When my kids rinse the dishes, they're not as clean coming out, and we spend more time cleaning spaghetti noodles out of the filter in the bottom of the dishwasher. Well, I think we're increasingly learning that Protein A columns are a lot like dishwashers. Protein A technology is clearly an amazing tool in our downstream process, enabling the achievement of 98 or sometimes as high as 99% purity in a single step. But I think we all know Protein A is not perfect. In fact, the residual contaminants that co-elute with our products usually require at least one, more typically two, and sometimes three polishing chromatography steps to clean up. So let's look quickly at the root causes for co-elution of impurities from the Protein A column. Of course, the root causes are imperfect and non-specific adsorption events that take place. Impurities can non-specifically adsorb to Protein A or to hydrophobic or charged residues on the resin surfaces. Advances in state-of-the-art Protein A resins have gone a long way toward eliminating these resin imperfections. But recent investigations of impurity co-elution really highlight the importance of so-called hitchhiking interactions. That is, interactions between impurities like HCP and DNA and the monoclonal antibody product itself, leading to co-elution of those associated impurities with the product. In fact, Recent studies suggest that certain subpopulations of host cell proteins may be particularly problematic with respect to these hitchhiking associations, as well as DNA protein complexes like chromatin. This type of interaction can't be eliminated by further improvement in protein A resins. By design, the monoclonal antibody is going to be present on the column at very high concentrations. So a very good motivation for performing chromatography upstream of the protein A column is to improve its performance by loading fewer impurities onto it. Additionally, we know that column lifetimes are ultimately limited by the aggressive cleaning procedures, like sodium hydroxide clean-in-place procedures, that we need to use between batches to remove column-bound impurities. Just as rinsing the dishes better reduces the need to empty the dishwasher filter, loading fewer impurities onto the protein A column may enable a reduction in the frequency of harsh cleaning procedures. This could substantially extend column life.
These are very compelling benefits, John. Would this also impact the customer overall process economics? And do you have data which can illustrate this benefit?
Absolutely, Himanshu. We and a number of our customers are beginning to collect quantitative data about the effect of better upstream purification on process economics through better protein A column performance and protection. Here's an example study we did in our lab where we clarified a CHO centrate with either a conventional depth filter or Emphaze AEX hybrid purifier, then loaded and eluted the clarified fluid from the protein A column, and finally did an aggressive clean-in-place procedure. We measured host cell protein levels at each of the number positions in the centrate, the clarified fluid, the protein A eluate, and the cleaning fluid. In this case, Emphaze AEX hybrid purifier achieved 24% HCP reduction during clarification versus 6% by the conventional depth filter. But this apparently modest difference resulted in a dramatic difference in product purity post-protein A. As you can see here, the HCP concentration in the protein A eluate was 19 times lower in the case of Emphaze AEX hybrid purifier. Similarly, the HCP concentration in the clean-in-place fluid was 17 times lower in the case of Emphaze AEX hybrid purifier. That represents a significant enhancement of protein A column cleanliness, suggesting that compared with conventional clarification, an equivalent level of column cleanliness might be maintained with a reduced frequency of the harsh cleaning procedures that degrade our columns. Himanshu, that could potentially significantly extend protein A column lifetimes. We are currently working with a number of our key customers to quantify this significant economic benefit. This is a similar study we did in which we looked at DNA at each of the same process positions. The Emphaze AEX hybrid purifier reduced DNA by 4 logs during clarification. As I said earlier, this is a typical reduction value for DNA. In this case, there was no measurable DNA reduction by the conventional depth filter we used. Again, contaminant reduction upstream of the protein A column paid big dividends in terms of both its performance and its cleanliness after elution. The DNA concentration in the protein A eluate was 3.5 logs lower in the case of the Emphaze AEX hybrid purifier, only 57 picograms per milliliter. In fact, as you can see by comparing the bars at positions 2 and 3, the Emphaze AEX hybrid purifier filtrate prior to protein A had a substantially lower DNA load than the conventional depth filter filtrate after protein A. Similarly, the DNA concentration in the protein A column cleaning fluid was nearly 3 logs lower in the case of Emphaze AEX hybrid purifier. Again, this suggests there is a significant opportunity to reduce the application of harsh cleaning procedures, particularly within a harvest batch, while maintaining a high degree of column cleanliness.
This is very exciting stuff, John. The potential would have a high impact on customer processes. Can you give example about how customers are using Emphaze AEX hybrid purifier?
Well, customers use Emphaze AEX hybrid purifier wherever they would otherwise use a fine grade of a conventional depth filter. For customers using centrifugation, the Emphaze AEX hybrid purifier can often be used to provide a single stage of clarification right after centrifugation. As I showed you before, the Emphaze AEX hybrid purifier provides throughput similar to a fine grade of conventional depth filter, and customers will realize all the benefits I talked about, particularly better performance and protection of the protein A column. As a secondary benefit, since the Emphaze AEX hybrid purifier contains a built-in 0.2 micron bioburden reduction membrane, customers can expect to require smaller surface areas of a downstream sterilizing grade membrane. In fact, we would strongly recommend using the superior particle reduction capability of the Emphaze AEX hybrid purifier to replace the 0.2 micron sterilizing membranes, that typically protect our protein A columns with even more protective 0.1 micron sterilizing membranes, like our recently launched LifeAssure PDA 010. This will enable mycoplasma level protection of the protein A column. Lately, many of our customers have had success replacing the centrifuge with a complete all single-use harvest and clarification process, typically by using a two-stage depth filter train. In that case, customers will have success replacing the second stage of conventional depth filters with the Emphaze AEX hybrid purifier. Together, the Emphaze AEX hybrid purifier and LifeAssure PDA 010 can provide a step change improvement in protein A column protection, including protection of the downstream process from soluble components that can only be robustly removed chromatographically, as well as mycoplasma that can only be removed by a 0.1 micron membrane.
John, can you summarize the value Emphaze AEX hybrid purifier will deliver to customer processes?
Sure. To summarize, the Emphaze AEX hybrid purifier is a new, all-synthetic clarifying product for biopharmaceutical purification. It brings to clarification an unprecedented level of anion exchange capacity, enabling flow-through chromatography during clarification to remove not only particles and cell debris, but also soluble impurities like DNA and host cell proteins not conventionally removed until further downstream. It is also the first ever clarifying product with a defined outlet pore size, providing guaranteed removal of small particles, including greater than 6 log bioburden reduction, and enabling protection of a downstream unit process from mycoplasma using a 0.1 micron sterilizing membrane like LifeAssure PDA 010. Keep in mind that even though the Emphaze AEX hybrid purifier is designed to work upstream of the protein A column, its substantial benefits are realized downstream of the protein A column, where the better performance of the protein A column provides substantially higher product purity upon elution. Recall that I showed you data in which clarification using Emphaze AEX hybrid purifier enabled the achievement of 19 times lower HCP and 3.5 logs lower DNA concentration in the protein A eluate versus conventional depth filtration. So the ultimate goal of any evaluation of the Emphaze AEX hybrid purifier should be to demonstrate its value in higher product purity downstream of protein A, as well as the opportunity to extend protein A column life. Those studies are involved, and so they probably won't be the first evaluations conducted by customers. But we strongly recommend monitoring the DNA and HCP reduction by Emphaze AEX hybrid purifier during any first test evaluation. With an optimized lab scale clarification train, customers should expect to observe a throughput of Emphaze AEX hybrid purifier similar to a fine grade of a best-in-class conventional depth filter. The ability to use a downstream 0.1 micron sterilizing membrane economically and with confidence, about 4 logs of DNA reduction, and about 20 to 40% HCP reduction. Once those observations are made, there's every reason for our customers to expect really exciting results from protein A column studies, both in terms of the protein A column's performance and its cleanliness.
So as we summarize, Emphaze AEX hybrid purifier removes about 4 logs of DNA, 20 to 40% of HCP, delivering better performance in protection of protein A column and leading to overall improved process economics. John, where can people find more information about the product?
Well, there's lots of valuable information for our sales representatives in the sales guide for Emphaze AEX hybrid purifier. For detailed information about everything we've talked about today, I'd also like to highlight the application guide. The application guide was designed to help our technical service folks, our sales folks, and our customers work together to evaluate the benefits of the Emphaze AEX hybrid purifier in our customers' facilities. It includes detailed descriptions of the product's design and benefits, detailed guidance on the application of the Emphaze AEX hybrid purifier in various common scenarios, detailed operating instructions, and a large number of case studies providing specific examples that quantify the benefits we've been talking about. We hope this information will help our technical service and sales team members and customers work together to discover the exciting benefits that can be realized by applying advanced materials science to clarification.
Well, thank you very much for your time, John.
You're welcome.